we say with our eyes? I wonder what happens in the eye. I wonder.
Welcome to the lecture about vision. It will be probably the first of 3 or 4 lectures on this topic, and today we'll talk about the eye. So let's start with an exercise. Take a piece of paper, find a pencil, and try to sketch the schematic of what is in the eye, all the parts. You know, this cross-section picture that they have in textbooks. It will help to tune into the lecture, and then we'll continue. Pause the video, do a sketch. Let's see, what do you remember? I'm guessing there is the eyeball, it's supposed to be roundish. The light is coming from the left, and it works kind of like a camera, right? The eye, in the sense that there is a lens somewhere over here. The lens is supposed to be transparent, and it's hanging on something. Something has to keep it in place. It's a cross-section, it's not a three-dimensional picture. What else do we have? On the other side, there have to be something that actually detects the light. It's called retina. So let's draw something here, and there will be some cells here. We'll talk about the retina later. So at the very least, if there is an object here, a human, that would be human. This object will create a picture. The light from this object will travel, and whatever light goes along the axis will basically just hit the retina in a straight line. And whatever light is coming at the angle will be focused, and you will have a picture on the retina. What else do we need? The retina needs to talk to the brain, so at some point somewhere there have to be a nerve, an optic nerve, that would send information to the brain. Another thing is the protective layers, cornea over here. The cornea is the part that faces the environment, and there are tears, so there have to be some tear glands, and you blink and clean it. And if you're running around or biking around and a fly runs into your eye and it hurts, that's the cornea that is sensitive. It has to protect your eye by making you care about it. Cornea can repair itself a little bit. There's the eyelid somewhere, and there would be eyelashes. I'm not drawing that, but theoretically they exist. What else? You know, there is a pupil, right? A pupil is a hole in the iris, so we need an iris, and the iris is not transparent, and the hole in the middle leads into the eye. Why do we have it? Why do we have the iris and the pupil? The size of the pupil can change. If we are in the dark, we need more light, so the pupil opens, and during the bright light of the day it may close a little bit and make the pupil smaller. Okay, so in order for that to happen, we'll need some muscles somewhere over here. I will draw them really approximately. What about the lens? Are there any muscles involved with the lens? Yes, because you know, you can look at something that's close to your eye, or you can look at something that's far away. You need to change the focus in the same way in which the camera can focus on things that are closer or things that are further away. If you're staring at the horizon, then all the objects that are close to your face are blurry, and the other way around. How do we do that? We have some muscles that connect to the lens that make this lens more round. We change the shape of the lens in order to focus on different objects. Okay, that's good. What else is missing? I guess one last thing we could draw are the muscles to rotate the eye itself. You can look to the left and you can look to the right and up and down. So there have to be some muscles that do that. They come in pairs. If you have a muscle that would rotate the eye upwards, there have to be a matching muscle that, if contracted, would rotate the eye downwards. So you'll have a pair for upwards, downwards, and then you'll have a pair for left and right. So it's 4 muscles. Actually, it's 6 because we have one more muscle that rotates the eye kind of like a camera. If you slightly tilt your head, the world doesn't tilt as much as it would tilt if you tilt your phone. For a while, it stays kind of more or less upright. For small movements, the eye itself rotates. There are 3 pairs of muscles connected to the eyeball. Alright, good. That's enough to at least start the conversation. Let's talk about the iris first. And that's a nice part because it has this pupillary reflex. If you're in the dark, your eyes become larger, the size of the pupil increases. If you're in the light, it becomes smaller. And you can even notice that if you stare at the mirror. If you turn the lights on while looking at your own reflection in the mirror, you can literally just see your pupil suddenly becoming smaller. If you're scared, if you're excited, it also changes its size. There are even people who try to track the size of the pupil in order to figure out what emotions a person experiences. Like people in advertisement show you one brand, they show you another brand, and measure your emotional reaction to that. I'm not quite sure how believable valuable these results are, I'm kind of naturally hesitant when something as simple is presumed to reflect your inner state, but it's interesting. And the way it changes its size, there are some muscles that run in a ring that make it smaller, and there are some muscles that run like a star that make it larger. They kind of work antagonistically in the same way in which muscles in our limbs work antagonistically. The idea is the same. And then goes the lens. And the interesting question is, what is the lens made of? Because you see, the lens is supposed to be of high optical quality. Of all things in the eye, it's one of the most important, arguably. Without a good lens, you won't have a good image. So the lens has to be really transparent and clear. But the problem with the body is that we are made of cells, except for teeth and nails that are synthesized by cells. And while a few small cells are fairly transparent, as you can see from the fact that our hands are not transparent, for example, or like if you've ever seen meat, it's not transparent. Just a bunch of cells won't give you a transparent tissue. We have again a little bit of a problem. So how can you make a lens if it's made of cells? Let's imagine that you are in an Archangel Gabriel construction bureau. You know that you need to develop a lens, but you have this technical constraint. What would you do? What would be your guess about what the evolution did? Pause the video, think a little bit, then unpause and compare what is the lens made of. Alright, what's your answer? How to make something super transparent, but also keep it alive? There are different ways to answer that, and one way would be to, okay, let's just synthesize the lens. Let's not make the lens out of cells. Let's do something like nails or teeth. Let's just make something transparent and let it be. And it is a legit approach, and some animals do that, but that works best with really small eyes, because it's hard to synthesize something perfect and precise if you're secreting it outside. What happened in our eye is actually different. It's made of cells, but the cells are practically dead. They're not completely dead. It's a weird story, and it's not neuroscience, but I think it's so amazing that I really want to talk about that. So this is a cross-section, and it looks kind of like an onion, but only in that cross-section. If you make a cross-section perpendicular to the flow of light, like almost like a cornel cross-section, then you see a bunch of irregular hexagons. These cells are not like layers in the onion, but more like columns. But the really interesting thing is that they don't have anything inside them except for this transparent stuff. They don't have DNA, they don't have a nucleus, they don't have any machinery to produce proteins. How come they appeared? How did they grow if they don't have any of that? Can a cell grow without DNA? Of course not. So what happened is that when we were in the womb, when we developed as an embryo, these were normal cells with normal nuclei, with DNA and everything, and they just kept growing and producing this crystalline-like transparent substance to fill themselves, to make a nice lens. They also packed themselves with some protective proteins that would almost like pickle them, not exactly pickle, but keep them preserved for the duration of your life. And then, once they were ready, they disassembled all the machinery that keeps cells alive. They disassembled the mitochondria, all the stuff that produces protein, and then in the last act of self-sacrifice, they get rid of the nucleus, so there is no DNA left. You can see that on the sides, there may be some cells that still retain nuclei, like here and here. But in the middle, all of them are now just empty husks of a cell, dead. So again, the same thing that I mentioned with ossicles. In order for you to see, some cells had to sacrifice themselves. That's how organisms work when you transition from the level of cells to the level of a multicellular organism. They behave almost altruistically in the sense that they sacrifice their life for the life of the organism as a whole, because that's the only way for them to propagate themselves. Because another organism, like the baby of this one, will appear and it will have the cells again or something, and it will have another lens. It's interesting if you think about it. Kind of weird. Okay, next. So we have the lens, we have a pupil, and then inner side of the eye and the retina. And the retina has layers, and some of the layers are obviously to catch the light, actually to detect it. So there have to be some detector cells, some receptor cells, photoreceptors. But then the retina also has some neurons in it, because at the very least it needs to send information to the brain. Right? So what layers are there? How does the light hit them from the very first layer that it meets to the last layer? Which is the first layer that the light hits? What do you think? Okay, this is a little bit of a trick question, because functionally the retina has to have receptors and then maybe some cells that listen to the receptors and do something with the signal that comes from the photoreceptors, and then there have to be a neuron that sends information to the brain. So functionally everything starts at receptors and then we have some neurons. But if you look at which layer is hit first, that's actually not the receptor layer, the layer of neurons. So the light, as it comes to the retina, first goes through a whole bunch of fibers that eventually leave the retina and go to the brain. And then the light goes through a whole bunch of neurons, and then through some other neurons, and then through some other neurons, and only at the very end it reaches photoreceptor cells that actually detect the light. This makes no sense if you think of that. That's wild. That's not what you would have expected, right? If you try to again play Archangel Gabriel and reverse engineer the eye, you would think, okay, the light is coming, so let's detect it with some receptors, and then let them talk to some neurons, and maybe they will talk to some other neurons, and they would send information to the brain. That's how it should be, but the thing that we have is the reverse of that. First neurons talking to the brain, then some other neurons talking to these neurons, and then the receptors, the last. That makes no sense. And it doesn't make any sense. I'm not tricking you now. I'm not saying that, oh, grand reveal, it's actually so nice. No, it's not. It's stupid. It's a blunder. The Archangel Gabriel Construction Bureau have totally failed us. Our eye makes no sense, this aspect of it. Like the pupil is brilliant, the lens is awesome, but the sequence of layers in the retina has no excuse whatsoever. It's stupid. And it's not like it's inconsequential. It has lots of consequences that we will now list. And it's not like there is some problem with creating the eye correctly, because there are totally animals that have correct eyes. Octopuses. So look, on the right, we have an octopus eye. We have these fibers that go to the brain. So we have the optic nerve, and then each fiber travels and connects to the cell, the receptor cell. So the topology looks kind of like that. But in our eye, everything is reversed. The fibers approach cells from the incorrect direction. They approach the photosensitive cells. The connections enter the retina from a wrong direction. They enter the retina in such a way that the light has no other option but hit the nerves and the neurons first. So it gets scattered and occluded, and the image is just worse than it could have been. Octopuses, check. Humans, fail. This is just a wrong, suboptimal, inefficient design, because it's not a design, but we'll talk about that. So are there any consequences to that? Of course there are, because again, the light entering the eye, instead of hitting the receptors and being detected, it's first getting scattered and refracted and distorted by these layers of neurons. And these layers may be relatively thin, relatively transparent, but it still damages the image. So first of all, because we have this weird situation, our blood vessels also come on top of our receptors. This is the image that a doctor sees when they study your eye. If you've ever been to an eye doctor, they give you some special drops. They make your pupils as large as possible, and then they use some machinery to stare into your eye, and that's what they see. That's how the retina looks through a doctor machine. And the most striking part of that is that it's filled with vessels. So not only the light is scattered by all these neurons that treacherously lie on top of your receptors, but also there are vessels, blood vessels, literally filled with blood cells traveling through them, that also obviously scatter the light. And in the description there is a link to a mini lab, not my video, very good video, that describes how you can see these blood vessels, because normally you don't see them, but there is a special trick using a piece of paper and a careful motion to see them, not the same way as the doctor sees them, but they will appear in your field of view, kind of in this shadowy form that is shown on this picture. But I won't describe it in detail. If you can afford to do it now, pause this video and go and run this lab. But even worse than that, not only we have all these vessels on top, we also have a blind spot. What is a blind spot? Oh my god, look at that. If you have a retina And for every receptor, there has to be a fiber that comes from it, and it comes from the wrong direction, from the inside of the eye. Then there has to be some place where all these fibers enter the eye. Or rather, I guess the better word would be to leave the eye, because ultimately information travels from the eye to the brain, not in the other direction. There is a place where all these fibers have to cross. And if these fibers cross there, then there are no photosensitive cells in this part. It's only the fibers. And indeed, if you look at the retina, there is this part where everything leaves the eye. And then we also have blood vessels. And okay, this place is wasted already. It cannot perceive light, so the blood vessels also enter and leave through this part. Arterial blood comes in, makes a tree of capillaries, and then venal blood deoxygenated, because blood brings oxygen and glucose, right? Food and breathing stuff. So it comes, distributes oxygen, then capillaries collect into veins again and leave through the same part. The net result, though, is that we have a blind spot in our retina. And if you haven't quite grasped the consequences of that, it means that right now, as you watch this video, there is a part of your visual field where you're blind. Like right now, in the part of your visual field, you're blind. You don't perceive anything with this part of your retina. And it's not like on the side, it's not somewhere on the periphery. As you can see, it's not like the fibers are coming from one side. No, they're coming relatively in the middle, not in the exact middle. The exact middle is kind of here. That's where the light hits if you just stare straight at something. But it's fairly close. It's not that far from the middle of from the point at which you're staring. So how come you don't see it? How come you don't realize that you walk around with this blind spot almost in the middle of your vision? What do you think? How come we don't usually notice it? The brain. The brain is smart. Thank you, brain. The brain is hiding this fact from you. Some circuits in the brain, before this information even reaches your consciousness, just hide the fact that a part of your visual field is missing. It is filled in, the official word for that. Some circuits, some neural circuits early in the hierarchy of processing visual information, just make a guess about what color, what amount of light, maybe even what patterns would be in this part of your visual field. And they hide it from you by pretending that they're really there. There is a way to see the blind spot, though, using this exercise. The way it works, you need to close your left eye, assuming that your right eye sees. Close your left eye and then try to place your head, your face, against this picture and stare at the circle on the left, the one that has R letter in it, like you're using your right eye to stare at the circle with the letter R. And then start gradually approaching it. And don't change where you look at. So like, don't do that. Don't move your eye, but just stare at the circle. But pay attention to this cross that you should see at the periphery. Does it make sense? It's kind of weird. You need to stare at one place, but pay attention to another part of the visual field. Pay attention to the periphery of what you see at the periphery. But if you do that, then at some point the cross will disappear. It just won't be there anymore. If you move a little bit further, it will appear again. If you move a little bit further apart, back in the original direction, it will appear again. But somewhere in the between, there will be some range of positions where you won't be able to see the cross. And you can do the same thing with your left eye, just you need to reverse everything. You need to stare at the cross and pay attention to the disappearing circle. It also works. You know what's going on, right? As you change the position of your head, if you have one eye and you stare at two pictures, the part you're staring at is always projected on the center of your visual field, but the cross will be projected further away if you bring your head close, or closer to the center if you move your head away from the image. And so somewhere over here, somewhere in between, is the blind spot. And so by moving your eye relative to the image back and forth, you can find a place where the image of the plus sign will be projected on the blind spot, and so you won't be able to see it. Or like the other way around with a circle if you use your left eye. If you haven't tried that yet, try that. Just pause the video and use this image to locate your blind spot and enjoy the fact that indeed you're blind there and your brain just fills it in this case with white background. Then let's go further. Why don't we see the blind spot in the vessels? Because the brain, right? And it turns out that the same thing happens even when people go partially blind, which is interesting. If a person has a problem in their retina, for example, retinal detachment or something, that's, by the way, why one shouldn't play with sticks too much, because they can harm their eyes and cause a retinal detachment. If part of the retina stops functioning, at first you see it as a black spot. At first, because there is no reporting from this part of the retina to the brain detects it as a no light zone, as if a person always has this black spot that moves with the direction of their gaze, right? If you look to the right, it's still in the same place. If you look to the left, it's still in the same place, as if it was glued in front of you. Regardless of where you look, you always see it. But then, if you wait for a few days, weeks, or months, at some point it's no longer noticed. The vision doesn't come back and the person is still blind in this spot. You can show them letters in this particular spot, ask them to read this letter, and they won't be able to do that unless they change the direction of their gaze. But it's now filled in. The brain starts to guess what color is there, what pattern. It's usually perceived as the blurry patch, and people notice that objects, when they cross into this patch, kind of cease to exist, as if being swallowed by a hole, and then if they cross further, they appear again. They like pop out of existence and then pop into existence again, but at least it's no longer perceived as a black shadow. Why is that? Why do you think this is happening? Like, which why is it? Kind of what is the benefit of having that in the brain? Is it just a side effect of our processing, or is it actually helpful? What do you think? Try to answer this question. Why would filling in be a thing? You have an idea? I guess the pictures on the left here look more annoying than the pictures on the right, especially this one with trees, where they show a little bit of pattern being recovered, being guessed by the brain correctly. It's less annoying when you don't have a black spot. You still don't see, but at least it doesn't look like an object. It looks like nothing to you. And in this case, nothing is a better representation of a blind spot than a black shadow. So the brain tries to cancel a stimulus if it's always there. Remember, we talked about that. We talked about that when we discussed smell. If the smell is always there, it's no longer informative. You don't want to be able to smell your own nose, because it doesn't make sense. Your nose is always there. So this smell shouldn't be a part of your sensory landscape. And in the same way, if you're in the room and there is always a noise in the room, you'd rather cancel it out. It would actually benefit you. It will help you to concentrate on sounds that make sense. And kind of the same thing happens here. If you think of all these blood vessels, why don't we see them? All the blood vessels that are supposed to form this spider-like network on top of your visual field? Because it's useless. It's a strange kind of teleological. It's philosophically not a sound way to answer questions like that. But you know what I mean, right? It is better if you learn to ignore them, not actively ignore them, not like take an effort, but if you learn to kind of compensate for their presence and negate their existence, you will be able to pay more attention to the world around you, which means that whatever animal learns to do that will have a benefit over the animal that needs to navigate the world through this spider-like network of blood vessels encroaching on the picture that the eye generates. So that's one consideration. It actually confers an evolutionary benefit if you learn to ignore your blind spot. But also, ignoring something is fairly easy for a neuron, as we talked about. If a neuron receives a constant activation, the neuron can just stop listening to that. It's relatively easy. Filling in is much trickier, but you can kind of imagine that it should be possible. Gather information from around and kind of average it. We haven't yet discussed how any of that works, but at least intuitively it feels like, if you can tell a squirrel and a chair apart, then for sure you can average visual information and just substitute it for the part that's missing. It feels like a simpler task. And a tiny tangent here. You can sometimes see the blind spot in another funny way. When you're making photos with a flash, you sometimes have this red eye thing. It turns out that sometimes if the person is not staring directly at you, but they're looking a little bit off, you can also see a white eye, yellow eye in one of their eyes, like in this picture. See these two people, two, not even one, but they look at the same part of the world somewhere to the left from the photographer, and they are at about the same distance from the photographer. And then as the flash goes off, they have a red eye in one of their eyes, but a white eye in the other. Can you think what's going on here? I won't give you an answer, but you can think about it at your leisure. Think about the blind spot and where it is and the geometry of this scene, and hopefully you can figure out why they have a white eye and why they have it in one eye only, not in both eyes. Next, okay, see, so many troubles, so many problems. The blood vessels, a whole blind spot, a whole piece of blindness that we always carry with us. All consequences of this bad design in how our eyes are constructed. Like, how is it possible? Why evolution created our eyes in such a suboptimal shape, especially considering that octopuses have this beautiful design? Like, why don't we have it? How come we got the wrong end of the stick? And the problem with this question is that we don't actually know, because all vertebrates that have reasonable eyes, not just mammals, but birds and lizards and snakes and fish, all vertebrates have this incorrect, suboptimal, imperfect, wasteful, hurtful, unnecessary design of their eyes, while many invertebrates have eyes that are proper and beautiful and well constructed. We don't have access to those ancestors of first vertebrates to see how the eye could have developed this way. And eyes, because they're soft, they don't make very good impressions in the fossil record, as you can imagine. But it looks like the problem here is that for our ancestors long ago, before even fish were a thing, when these proto-vertebrates looked like basically a worm with a piece of cartilage in the middle, it looks like somewhere at this point the eyes have developed, and the eyes were so tiny and so simple that it didn't matter where the fibers would be coming from. If your eye literally looks like a little chamber with a hole, something like that, or maybe even that, if there is no lens yet, it doesn't matter actually where the fibers are coming from. They can come from here, as it is kind of shown in this picture, but they can as well come from here. It doesn't matter, because the quality of the picture doesn't suffer, because the quality of the picture is extremely low in this case. And the eyes of this shape are a legitimate stage in the development of vision, and vision developed more than once in the course of evolution, by the way. Vision is so nice to have that it developed independently several times, because it's relatively easy to make. You just have some cells that are photosensitive. It turns out that chemically it's relatively easy. And typically what happens if you have a worm, it's nice to have a bunch of photosensitive cells, because then you know whether the light shines on you or not, and then you can hide if it shines on you. That's what you use vision for when you're a lowly worm. Or maybe you go towards the light or you go against the light, depending on what you eat. But this is not image forming, right? The next stage is something like that, when you have a little hole and you have cells around it, because the light coming through this opening will only hit 1/2 of the cells. We kind of talked about that when we discussed infrared vision in pit vipers. Remember, they have pits, and these pits don't have a lens, but still having a pit is enough to make some sort of a very lousy image, because if the source of the radiation is over here and the hole is small enough, only cells on one part will be activated. So it's like a proto-retina. In case of pit vipers, it's actually skin and heat receptors, but in case of normal photoreception, these are some photoreceptors, just cells sensitive to light. But in an eye like that, in a very primitive eye like that, it doesn't matter where the fibers are. It doesn't matter whether they come from here or from here, from the inside of this bit. This is the basic idea that people have about why our eyes are so lousy. Early on, it didn't matter, and then it was too late to change. Here's a metaphor for you. Let's say we have a medieval town, and in this medieval town, you need to explain to a pilgrim how to reach from the entrance to the town, which will be in the left bottom corner over here, to the library. how to get from here to the library. And this set of rules for finding a library is kind of similar to rules of development as the organism develops. If you think of axons of neurons growing from one part of the body to another, or of tissues organizing, setting the limits for organs. We'll talk more about that, but in spirit that's pretty similar. So let's say the pilgrim starts here and the library is here in the nearest house over. So the rule is pretty simple. Turn left, here's the library. Done. So far so good, it works. Let's say then evolution happens and the species changes a little bit. The organism of members of the species becomes slightly different. They change shapes, some new organs appear maybe, or something changes about them. So now the library is over here in the next building over. Will the old rule work? No. If you just turn left, you don't see the library. But with just a slight variation, we can make it work. So the new rule that works will be turn left and go. Turn left and go and check every building on the right. Will this building work? No. Will this shed work? No. What about this one? Yay, it worked. So the rule now goes like that. Turn to the left, go, check buildings on the right until you find the library. And this rule is pretty robust because slight changes in the shape of the organism, of the species, slight variations won't ruin it. So let's say the library moved over here. Will the rule work? Yeah. Turn left, go, library, nope, 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 nope. Yay! We reached it. See, it still works. Let's say now the library moved over here in this big building. Will it work? Yes. No need to change the rule. Go, 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 check on the right. Yes, we came to the building. What if the library moved over here? Now it's something interesting happens because the old rule doesn't work anymore. If you just start and then you go forward, you'll go like in this direction, you won't reach the library ever. The library is here. However, again, one tiny mutation if it happens, and the mutation is, except for the first intersection, at big intersections, turn right. If you add this new rule into the system as a mutation, it now will function because like this is the library. We turn left, we go, 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 checking buildings on the right. None of them is a library. We reach a big intersection and then we turn right. We turn right at intersections. We keep checking buildings on our right. This one, nope. This one, yay, we came to the library. See, it still works. Now, what will happen if the library moves over here? Will the rule work? Yes, it will still work. You turn left, you go, checking buildings, none of them matches. We get to the intersection, we turn right. We get to another intersection, we turn right. We come to the library. And it will work even if the library is here. But see what's happening? The rule will work, but the way it will work, we'll turn left, we'll go all the way here, reach the first big intersection, turn right, reach the second big intersection, turn right, no intersections until we find a building on our right, that is the library. And at this point, it's really, really inefficient. A simpler rule, go forward, check on the left, would have worked better. We would have had a shorter path, but this mutation is really unlikely. It's really unlikely because in this case, even in this simplest example, two mutations should happen simultaneously. In all previous improvements were always one mutation worth. Like now add this rule, now modify this rule a little bit, you know? But in order to switch from this red path that I drew to a shorter blue path that would go like that, you need to change not one, but two things. You need to turn right on the first intersection, go forward instead of turning left, that's one. But then also you need to check buildings on your left side, not on your right side. And the probability that these two mutations will happen simultaneously is much lower than the probability of one mutation happening. And see, if either of these mutations happen alone, it will ruin the path. If you go straight but keep checking on the right, you never reach the library. You go like you leave the town. If you go to the left, but you keep checking buildings on the left, you also never find the library, even if you keep turning right, because the library is not on your left side. Does it make sense? The only chance to change from this path that works to this new path that's better is for two mutations to happen. And in real life, it's never two, it's like 10 or 20. So if something works, don't break it. That's essentially how evolution functions. Evolution is a tinkerer. It can improve on the existing solution, but it never gives up on the existing solution because this temporary point when the old solution doesn't work anymore, but the new solution is not yet functional, is tremendously disadvantageous. You cannot temporarily have the entire population go blind just to evolve new eyes from scratch. At least not under normal conditions, right? Maybe something like a mega super extinction, a giant extinction event, a giant volcano killing everything on Earth, or a giant asteroid crashing into the planet. Events like that could potentially wipe so much of existing solutions that surviving animals would have to invent everything from scratch. But under normal circumstances, with these incremental improvements, the chances of abandoning an old suboptimal solution are very low. You're much more likely to optimize and squeeze every last millimeter of optimization from the old suboptimal solution instead. And we will see this theme again and again in our discussions of brains, because brains are full of these optimized, highly optimized inefficiencies. Stupid solutions, unfortunate solutions that kind of work and look amazing, even though all this time evolution was kind of optimizing a wrong thing. It works, but it could have worked so much better if somebody could just make a step back and rethink what's going on and start from scratch. Never happens in practice though. So what happens in real life? What does evolution do in case of our eyes? It just tries to fix things. We try to make the best of the machinery that we have. And we already discussed one aspect. The brain is just dutifully trying to ignore all these disturbances to recover the picture, ignore the scattering of the light and the absorption, the blind spot and the shadows of the vessels that are always present. But also there are some other tricks. One of them is Müller glia. I wanted to include all these pictures just because they're so beautiful, but essentially, remember glia? Glial cells, like those cells that are also in the brain, but that are not neurons. We talked about astrocytes that help synapses to work. We talked about oligodendrocytes that do myelination. So that's another type of glial cell, and it does lots of interesting things, but one thing that it does, it's just being there and being really transparent. It's more transparent than your typical cell, and it has this funny shape, and it totally acts as an optical fiber. It catches the light where the neurons are. See, these guys here are the neurons, but instead of allowing this light to get scattered by the neurons, it tries to catch it and redirect it towards the photosensitive cells. And that's bizarre. We could have made everything so much easier if just the neurons would grow from the bottom and not from the top, but we cannot reverse that. So now evolution is trying to fix that in this weird way. But that's only the beginning of the fixing process, because this can help a little bit with sensitivity, but it's not gonna help with blur. You still, instead of having tiny receptors directly exposed to the image, have all this stuff sitting in front of them, even if this stuff is transparent. What can you do? Okay, fix number 2, a fovea. What is a fovea? It is a part of your retina that you use to stare at things, and this is the part that produces the most high-resolution picture, and it looks like a little hole in your retina, where the layers of neurons are moved to the sides and where there are no vessels, because all the vessels come close to it but then stop. So they try to saturate the fluid around the fovea with oxygen, but they don't cross into the fovea itself. And because of that, in the fovea, and only in the fovea, that is the only part of our retina where the light, as it comes from the lens, hits the receptor cells almost directly. If you look at the retina on this doctor image, here's the fovea over here. See how there are no vessels? All the vessels are coming to it but then stopping a little bit. They almost approach it. Here's the oxygen, but they don't cross into the fovea itself. There are no vessels over here, and there are no nerves over here at the expense of all these poor cells nearby, because they now have all this machinery, even more machinery than usual, sitting above them, like over here and over here. But at least those few cells in the fovea get a really nice image. And so that's what we use for high-resolution vision. When you look at this presentation and you can read letters, or you can see these pictures in detail, you use your fovea. For any aspect of vision where seeing details is important, this is the only part of the retina that allows you to capture the world in high resolution. And this is cool, but that's also weird because it's only a tiny part of the visual field. So imagine this is the entire retina, right? Which means that only one part of your visual field is in high resolution. You have the blind spot, of course, but everything that's around, everything that's not the blind spot, but also not the fovea, is in bad resolution, is really blurry. So we fixed one thing, but it still doesn't fix it completely. That's how you actually perceive the world. There is this tiny part that is in high res, but the further you go, the the more blurry everything becomes, because the receptive fields of different rods and cones, they just grow in size. So you cannot figure out anything within this area or within this area. And the further you go from the fovea, the worse it gets. On the right, you have this image of how it might look like actually if you look at an image like around here. This part, this only part, will be in high resolution, but the further you go away, the more blurry, the more pixely it becomes. Our pixels, kind of natural pixels of the retina, are of course not square, it's just a bunch of overlapping zones, but the gist of it is like that. Everything in the periphery is blurry. And you can kind of perceive that if you pick some high resolution image, maybe in your room somewhere, like find something and just stare at one point in the middle and try to concentrate on everything that you see with your peripheral vision. You will realize that you can see something, like you are aware of other objects that are in the periphery that are like over here or over here. You can figure out their basic shape, you can figure out their basic color, but you cannot, for example, see details on them. You cannot count buttons on a shirt if the shirt is on your peripheral vision. You cannot read a text on a poster if the poster is in your peripheral vision. And here's an example that I came up with. It is kind of a demonstration, so let's do it together. Stare at this dot, like look at this dot, look at it very carefully, and then I will press a button and some other stuff will appear on the screen. But your goal is to continue looking at the dot, but pay attention to the stuff around. So don't move your gaze, don't do that. Don't change where your eyes are looking. Keep looking on the dot, and while looking at the dot, try to figure out what's happening around it. Okay, ready? Look at the dot. Here, now we have some letters. So keep staring at the dot, but try to read the letters using your peripheral vision only. Can you do that? I hope you're looking at it on normal computer screen and not on the phone, by the way, because if you're doing it on the phone, obviously the image is too small and you cannot do anything with it. But if you're using a computer screen, then you should be fine. So can you read it? I hope you kind of can. I kind of can do it barely. And that's how I constructed this image. Okay, feel free to look at it now. I made letters that are further away from the center larger, to compensate for the fact that our vision becomes blurry. And because of that, even if you stare at the center, you should be able to kind of barely figure out what these letters are. But it becomes increasingly hard as you go away from the middle, even though in this particular picture, the letters become increasingly large as we go away from the middle. And if you think of it, this image is actually still fairly central in your vision. Like, we're not talking something really at the periphery, something really to the right or really to the left. It's still mostly in front of you, just not exactly in the middle, not exactly the fovea. So then the question, how come we don't realize that? How come when we walk around, if a person have never studied neuroscience, they will never ever realize that the world is only in high resolution in the middle of the visual field? When we look around, we kind of perceive this world as not blurry, as similarly crispy, depending on how good your vision is, of course, but still similar amount of crispness in all parts of it. We don't have this feeling that there is a giant blurry field that's moving with our gaze. We kind of look at the entire picture and we kind of see it. How does it work if actually, when we take an effort and not move our eyes, we can see crisp picture only in the very middle? What do you think? You know the answer, right? I hinted heavily. We move our eyes. In real life, we always move our eyes. And it is known for quite some time now. This is a picture from the 50s, and this is probably the most famous young Russian girl in neuroscience, because the dude who first recorded how we move our eyes when we look at the picture was Russian, and his name was Alfred Yarbus. He figured out that when we look at an image, we always do these tiny micro jumps with our sight. We never look at one point for too long. Several times a second, we slightly change the direction of our gaze, and we don't realize that usually. You can kind of force yourself to reflect on that and realize that, if you think of how you're looking at the image. Like if you try to look at this girl now, but you pay attention what part of the image you're looking at, you would probably notice that you're moving your gaze. But without this additional prompt, you would have just thought that you look at the picture in general, while actually you would be doing this search all the time. And he measured how you do that, like what part of the image are most informative. And it turns out that a normal human, when looking at a normal human face, typically looks at the eyes a lot, and then at the mouth a lot, and then also follows the counters and stuff like that. On the right, we can see about a minute or two, I'm not quite sure, of this changing the direction of gaze. And there are two words here, foveation and saccades. The process of jumping, the process of changing the direction of your gaze slightly is called a saccade or saccade. I think different people pronounce it differently. So every time you jump, it's called a saccade. Another name is foveation, which is bringing the image into fovea. If you're looking at something and then something interesting happens nearby in your visual field, you immediately stare exactly at it, and you don't even have to turn your head, right? You can just look in there, and that's called foveation. Say when you read, you have to do saccades along the text. You have to foveate again and again and again, so that you could actually read the letters, because you cannot read letters with your peripheral vision. And the story of this dude is amazing just because he did these experiments still as a grad student in 1948, and there were no computers back then. These days, it's very easy to do that. These days, you just put a little camera in front of the eye, and the camera tracks the position of the pupil, and then you somehow match it to the image, and you can figure out where a person looks like without any special devices. Like these days, there are literally studies where marketing firms, marketing groups track the gaze of customers that look at the shelves at the store. They don't actually do it with normal customers with the store, because that would be an invasion of privacy and everything. They have like a fake store, and they invite people from a focus group, and they know what they're doing, and they're being paid for that. But their job is basically just to look at the shelf while a camera tracks movements of their eye and figures out where they look first and where they look second and so on. But back then, in 1948, the only thing they could do was to glue a little mirror on the surface of the eye, and then they would sit in a really dark room staring at this image, while at the same time a light would shine on their eye from a different direction, get reflected from this tiny mirror and projected on a photo plate, so that every time the eye would move, it would leave a trace on this photo plate. If you think of that, I mean, it's so painful. It wasn't actually, the mirror wasn't glued to the eye, there was a suction cap, but still it was a pretty painful experiment, and he mostly experimented on himself. I think I read somewhere that when these experiments were published and translated into English, nobody believed him at first, because they thought it's impossible to build a rig like that. Okay, good, let's repeat that. We have a crappy retina, but then we created a fovea by pushing everything to the side. But then now we have a good picture in only one part of our retina, so we have to move our eye all the time. Now we run into a different problem. If we move our eyes all the time, how come we don't have this shaky cam effect? Like, you know, if you take your phone and you try to record a video, but you do this thing, like you change the direction of it all the time, the same way as our eye moves, you can imagine how this video would look like. If you ever watched on YouTube videos of some dramatic events, and at some point the camera person starts to run and everything is super shaky. These videos are typically very hard to look at, but our eyes do these changes in the direction of our gaze all the time, several times a second. And if you look at this image, you can also see that our eyes, unlike cameras that they use for movies, are kind of squishy. And they're not made of glass and metal, they're made of soft tissue, which means that every time we move them, and we move them really fast, they wobble. Do you see this wobble? How come we don't get dizzy of this image always shaking around? How to solve this problem? This is the question that you should be able to answer if you use your common sense really well. So pause the video, think about that. How come we don't see this horribly blurry image all the time and don't develop a headache because of that or something? Pause the video, think about that, then unpause, compare the answer. We go blind. That's the answer. We go blind. Every time our eye moves, we temporarily go blind. It's called saccadic masking. And if you do the math, it's bizarre because we go blind. We stop perceiving the image every time our eye moves, but our eye moves for up to 100 milliseconds, usually a little bit shorter, but let's say 50 milliseconds. And we do it several times a second. So let's say 2 times a second, and there are 1000 milliseconds in a second. We're blind 1/10 of time. This is a very approximate calculation. Like Wikipedia claims that we're blind 40 minutes a day, but it's somewhere close to 1 hour a day if you think of it. That's the order of magnitude. Not only we have a blind spot in space, just present in our visual field, also in time, about 10% of our waking time, we're blind in this tiny pieces of blindness that come and go, only to save us from these constant periods of blurriness that, if they reached our consciousness, would of course totally screw up any object recognition. But the blurriness only happens because of saccades, that only happen because we have the fovea, and the fovea is so tiny, which in turn only happens because our eye is inverted. Do you feel how weird it is? That kind of gives us a good taste of how evolution works and why my exercises about imagine yourself an Archangel Gabriel are useful sometimes, but crucially not always. Evolution is a tinkerer. That's a famous saying. So it's not an inventor, it's a tinkerer. You can, by tinkering, like by chipping a little bit here or adding a little bit of scotch, of duct tape here, you may like try to make it a little bit better, a little bit longer, a little bit flatter, a little bit redder, a little bit more transparent, smaller, larger, like bend it a little bit. And anything that's achievable through these tiny steps is achievable, but huge revamps are really hard and often impossible to do. And the more complex and beautiful and nicely looking and almost designed looking the structure is, the harder it is to remake it, because you have more to lose if you start messing with it. And our eye and our visual system in general is a great example of that. One crazy screw up early on at the stage of a lowly worm who just basically benefited from knowing whether it's being eaten from the back or from the front, at the stage of a lensless eye, an eye without a lens. One screw up at this moment, and then for millions of years, for billions of years, we're dealing with the consequences, and the ingenuity of compensations that cover this screw up are amazing. But think of an octopus. An octopus doesn't have to do any of that. It doesn't have to have a fovea. It can have good vision in all parts of the retina. And so it doesn't have to do saccades. And so it doesn't have to do this masking. It doesn't have to do circuitry for turning you blind, but then also hiding this fact from you, because you don't realize that, right? There are special visual illusions that show you that masking actually helps. And there is a beautiful lab, a really simple lab that I will encourage you to run. It's in the description. But the amount of work that goes into the cover-up with this early screw-up is just unbelievable. This concludes the first part of this video. Tune into the second part.
How do we see with our eyes? I wonder what happens in the eye. I wonder how does it even work? I wonder. Let us talk about